Hello students and welcome to the lecture of Microbiology, Paper 1, Introduction to Microbial World, Chapter 2, Golden Era of Microbiology, and the topic for today's lecture is Discovery of Viruses and the Reverse Postulates. Two scientists, Dmitry Ivanovsky, a Russian botanist, and Martinus Beijerinck, studied viruses individually. Dmitry Ivanovsky, who was a botanist, was studying the tobacco mosaic virus because plants of tobacco were infected with some virus which was unknown. So Ivanovsky tried to apply the Koch's postulates to the disease and tried to isolate the infectious agent. He took extracts of infected leaves and crushed the leaves in order to get the extracts from the infected leaves. The extracts that were obtained were then filtered through bacteria-proof filters, that is, the Chamberlain filter candles. Because these filters do not allow bacteria to pass through them, and thus they were referred to as the bacteria-proof filters. But even after filtration, the filtrate that was obtained was infectious. So Ivanovsky concluded that the particles or the agents that are responsible for the tobacco mosaic disease in the tobacco plants were certain filterable agents and not bacteria. Similar experiments were performed by Martinus Beijerinck. In 1898, Beijerinck performed experiments with plants infected with the tobacco mosaic disease. His experiments showed that the agent which was responsible for the disease was fluid and it was non-particulate. He noted that the pathogen was unable to reproduce outside the host and it seemed to multiply only in the parts of the plant that were undergoing a rapid cell division. He also observed that the filtrate free of bacteria retained the ability to cause diseases in the plants, even after repeated dilutions were made. He called the agent as Contagium vivium fluidum, that means a contagious living fluid. This was a revolutionary idea which was of course later understood to be the concept of a virus. So Dmitry Ivanovsky and Martinus Beijerinck had discovered the tobacco mosaic virus, although they could not see it because the microscopes in those days did not give larger magnifications to observe the viruses. As compared to the bacteria, the size of the viruses is very small. Bacterial cells like Bacillus subtilis has a length of 4 to 10 micrometers and a diameter of 0.25 to 1 micrometer, while the size of the virus particle is up to 0.018 micrometers. That is too small to be seen under the light microscope. But the two scientists had successfully had discovered the tobacco mosaic virus, and this was a milestone in the study of virology. But because the viral particle was not observed, people almost forgot the discovery of the viruses until in 1935 Wendell Stanley crystallized the tobacco mosaic virus, and then the virus was observed. After the discovery of virus that causes infection in the tobacco plants, a new virus was discovered which was called as the bacteriophage. Bacteriophage is a virus that causes infection in the bacteria. It was discovered independently in 1915 by Frederick Todt in Great Britain and in 1970 by Felix de Herelle in France. The study of bacteriophages was actually done because of the action of these phages or these viruses on the bacteria. The viruses caused the lysis of the bacterial cell, and therefore both the scientists referred them to be the lytic organisms or lytic. Frederick Todt noted that the bacteriolytic agent that he was observing exhibited two properties of the viruses, that is, the ability to pass through bacteria-proof filters and the requirement of cells, that is bacteria, for the growth of the particles or the lytic agents. So Todt thought that it may be possible that the bacteriolytic agent might be a virus, but he favored the possibility that it may be an enzyme that may be secreted by the bacteria that may be responsible for the lysis of the bacterial cell. In 1917, Felix de Herelle found that viruses were responsible for the lysis of the dysentery bacillus. De Herelle was quite sure that the phenomenon he observed of the lysis of bacteria was due to the virus which was able to infect the bacteria. So he coined the term bacteriophage and gave the name to the virus as a bacteriophage, that means eaters of bacteria. He also noted that the growth of the bacteriophage in the medium containing bacterial growth led to the appearance of clear zones in the film of bacteria on the agar plate. These clear zones were referred to as the plaques, and therefore the name bacteriophage was an appropriate name to the virus that caused the lysis of the bacteria. Felix de Herelle made certain notable observations about the viruses. The first he said was the bactericidal microbe cannot be seen under the light microscope or under the microscopes that were used by Herelle in those days. The second observation that he made was that the invisible microbe has antagonistic property against the bacillus of Shiga, that is isolated from the feces of patients recovering from bacillary dysentery as well as from the urine of other patients. The next observation that was made was that the invisible microbe can be isolated from patients only when the Shiga bacillus is present. De Herelle never found the microbe in healthy individuals. So he confirmed that the antagonistic effect of the antibacterial microbe is seen only in association with the Shiga bacillus. That is, the agent is a host-specific agent. He then declared that the agent is a living germ. That is, it can grow only in the presence of the Shiga bacillus. He observed that since the antagonistic microbe can never be cultivated in medium in the absence of the dysentery bacillus, it is an obligate bacteriophage. De Herel's next goal was to develop therapies against the infections that are caused by bacteria that could be based on the use of the bacteriolytic phages. He achieved his first success in 1919 when he stopped the typhoid plague in chicken by means of a phage that was isolated from the fecal sample of the typhoid infected chicken. Later, after the discovery of the electron microscope, a number of viruses and bacteriophages were isolated and studied in detail. Robert Koch in 1876 gave the Koch's postulates, which are important to correlate a bacteria to a specific infection. But at the time of Robert Koch, viruses were unknown, so he could not include viruses in his postulates. So later, in 1900, an army surgeon, Walter Reed, proved that viruses were responsible for yellow fever, which was caused due to certain mosquitoes, where it was observed that the mosquitoes were the vectors which carried the virus from one individual to another. So subsequent control of mosquitoes virtually eliminated the epidemic of yellow fever from Cuba and areas of Central and South America, and made it possible to complete the Panama Canal. Later, in 1937, Thomas River outlined the criteria similar to Koch's postulates to establish the role of specific virus as the cause of a specific disease. These postulates are nothing but modifications of the Koch's postulates, which are: number one, the virus must be present in the host cells or other body fluids at the time of the disease, showing lesions specific to that disease. The second postulate is the host material with viral agent used to inoculate the healthy host, that is the test organism or animal, must be free from any other microorganism. The third statement is the viral agent obtained from the infected host must produce the specific disease in a suitable healthy host and or provide evidence of infection by inducing the formation of antibodies specific to that agent. And the fourth statement is a similar material, that is the viral particle from newly infected host, that is the test organism or animal, must be isolated and capable of transmitting the specific disease to other healthy hosts. In the Koch's postulates, Robert Koch had isolated the organism in pure cultures and grown it in his laboratory. But with viruses, it was not possible to grow the viruses in the laboratory, and therefore, instead of obtaining the virus in a pure form, in material that is infected from the infected host is used as the agent to transmit the disease to the new or a healthy animal. These are the reverse postulates with which we can say that a specific virus is responsible for a specific disease in a specific animal. Thank you.